Hi everyone, welcome back to another video. Today's lesson is going to be on photosynthesis, and it's another installment in the biochemistry series. Photosynthesis is a very important topic to know for biology in general and for classes like AP and honors biology and for competitions like USABO. So, um, yeah, it's going to be a lot of free points usually on those competitions and tests as long as you know it well. So let's get started. Okay, so here is the table of contents for this lesson. The first one is kind of like the applications of photosynthesis in the ecosystem. Um, Part two is an overview. Then we'll go to the light reactions in the Calvin cycle, which are part of photosynthesis, and we'll look at alternate photosynthesis mechanisms last. So first is the role of photosynthesis in the ecosystem. So we'll introduce the idea of trophs. So an autotroph is a self-feeder, and they're also called producers because they make everything from inorganic raw materials from the environment. And they provide for non-autotrophs, which usually eat them or absorb them, and photoautotrophs, which are a subtype of autotrophs, make their energy from using light, and these include plants, algae, some protists, and some prokaryotes. And those are just some examples of some photoautotrophs. So heterotrophs are other feeders. They're also called consumers, and they eat other organisms or remains or organic litter of those organisms. So those would be decomposers. So if you look at this picture over here, they're just some examples of some autotrophs, some photoautotrophs, which are going to be the organisms we're focusing on today since they actually do photosynthesis. Alright, so part two is an overview of kind of how photosynthesis goes. So photosynthesis happens in the chloroplast, and chloroplasts are probably a product of endosymbiosis in which one organism engulfs another one. So the theory, which also applies to mitochondria, is that the sort of proto-eukaryotic cell engulfed a prokaryotic cell, and that prokaryotic cell essentially did work for that proto-eukaryotic cell, and together they kind of formed a relationship, and eventually the chloroplast became an organelle within the eukaryotic cell. So it's important to know the parts of a chloroplast. So just look at this chart. I would say that the most important things to know is that thylakoids, which are these like pancake stacks here, that's where your photosynthesis is going to be happening. The stroma, which is this fluid surrounding all the pancake stacks, that's where the Calvin cycle is happening. So, I think I said photosynthesis for the thylakoids earlier, but those are the light reactions that are happening there. And then stroma is for the Calvin cycle. And then you should note that there are two membranes of the chloroplast, and additionally the thylakoid membrane on the inside. So really three. And when we have a stack of thylakoids, we call that a grana, and plural is grana. And stroma lamellae are these kind of connecting pieces in between some of the thylakoids. So yeah, the chloroplasts are mostly located in leaves, especially in mesophyll cells, where there can be 30 to 40 per cell, and it's important that they get the resources that they need for photosynthesis, so they get this via gas exchange via the stomata. Okay, so here's just some chemistry stuff to look at to better understand photosynthesis. So here's the overall reaction for photosynthesis. It does overlook a lot of things that we'll be going into today, but this is just overall what goes in and what comes out. So photosynthesis is endergonic, meaning that we had to put energy in, namely light energy. So yeah, because if you think about it, photosynthesis, we're putting energy in so we can make sugar that we can later break down for energy. So here's the net equation of photosynthesis, and then this is what it looks like in the simplest form. So you'll be seeing this quite a bit. The brackets around CH2O, that just means some form of sugar because carbohydrates are going to look something like a multiple of this, and the brackets are just to indicate that it's not something, it doesn't actually exist. This is just what happens when you cancel out all the numbers, basically. Alright, so here's the history of kind of how photosynthesis has been viewed from a chemistry type perspective. So a scientist named Ingenhousz first theorized that this is how photosynthesis is going to go. So they had the reaction already, but what he thought was that this oxygen that's released as a result of photosynthesis comes from the CO2 that's been taken in. And we'll later find out that this is wrong. But Van Niel, another individual who contributed to this, was studying purple sulfur bacteria, who instead of using H2O used H2S. And what he found was that sulfur was released here, and this made it easier to see things because we have a lot of oxygens up in our normal photosynthesis equation, and that can make it confusing because we don't know which oxygen went where, right? But it's very clear from what the purple sulfur bacteria did that it's the sulfurs that were released. So that allowed him to come up with kind of a general form for this equation, in which, S and O could be anything. They just called it X. And yeah, that's what the general equation looks like. So this is a pretty big step forward for us realizing, hey, this oxygen here, it actually doesn't come from this CO2. So yeah, the C obviously goes here, H2 comes here, and the S is released. So Rubin and Kamen finally kind of cracked the code. They figured out, where the oxygens went, and the way they did that was they used a heavy isotope of oxygen. It's called oxygen 18, and then they tracked where that went, and basically this labeled oxygen, it was released when it was in the water, and then unlabeled oxygen, which is in CO2, that unlabeled oxygen went into the sugar and into the water. So yeah, this is the correct way for how photosynthesis goes, and it did take a while until we got here, but this experiment is pretty important to remember. Just know they used a heavy isotope of oxygen to figure out where each of the oxygens went, essentially. So yeah, here's just a quick summary of what goes where. All of the carbons in our sugar are going to come from CO2 because there's no other source of carbon. The oxygens in CO2, half of them go into the sugars, the other half go into water. And then, the hydrogens from water, part of them go into the sugars and another part go into water. And then all of the oxygen that's released comes from water. So yeah, here are the stages of photosynthesis. So in the light reactions, which is kind of like the photo part of photosynthesis, this happens in the thylakoid membrane. And in this, the big idea is we turn solar energy into chemical energy. So energy from the sun into chemical energy in the form of sugar. So what we do is we split water, and then that releases the O2 we talked about earlier, and that also creates some protons, some H plus ions, and it also creates some electrons. So what we do with these is we use them, we kind of like manipulate them and turn ADP into ATP and NADP plus into NADPH. So essentially we're making NADPH and ATP, which are sources of energy, And these go into the Calvin cycle, which is the synthesis part of photosynthesis. So this happens in the stroma, which, if you'll remember, is the kind of liquid around the thylakoids. And the Calvin cycle is often called like the light independent reactions, because it doesn't directly need light to function, because in the light reactions we needed light to split the water. That was that input of energy. But for the Calvin cycle, you only need the energy sources that you made in the light reactions. So yeah, it uses those energy sources to make sugar from CO2. And then when those energy sources are exhausted, they return back to the light reactions, where they get replenished for more Calvin cycle action. Alright, so let's move on to the light reactions. So first we have to understand kind of what is light. So light acts like a particle and as a wave at the same time, meaning that it has a frequency and a wavelength. And photons, which are units of light, have a fixed quantity of energy, and that's inversely related to wavelength. So basically, the shorter the wavelength, the higher energy a photon is going to be. And then this concept of light having a certain frequency is going to be pretty important in, the next parts of this lesson, because that largely determines kind of what wavelengths of light can be absorbed and used for splitting water. So yeah, here's just some pictures to help you kind of get more familiar with light and waves. Okay, so pigments, also a very important thing to know about. A pigment is a substance that absorbs visible light, and they're going to look like the colors they don't absorb. So let's say I have a blue pigment, that means it absorbs every single color except for blue, or at least it absorbs colors, and then the colors that it doesn't absorb combine and look like they're blue. So basically for our pigments, they have something called an absorption spectrum, and that basically is how much light they absorb of every wavelength. And then here it is for 3 types of pigments: chlorophyll A, chlorophyll B, and carotenoids. So it's going to look like that. And then the pigments a plant has and the absorption spectrum of those pigments determines the action spectrum of the plant. Basically, how much it can use certain wavelengths of light to do things, namely photosynthesis. So if you see this shape, it follows kind of like the shape you would get if you averaged all of these results out, because plants have all 3 of these pigments, and they usually would use all of them to help do photosynthesis. So yeah, this is chlorophyll, which is kind of like the big pigment. And chlorophyll A and chlorophyll B are remarkably similar, except for this one little group right here, which could either be CH3 or CHO. And yeah, there is a porphyrin ring with a magnesium in the middle. This is really important to remember because magnesium, it's important to plants for this purpose. If they start lacking magnesium, they're going to get chlorosis. Their leaves are going to turn yellow because they can't be, they're not making enough chlorophyll. So yeah, and the thing you use to create an absorption spectrum like this one here is called a spectrophotometer. But Engelmann, which was a scientist, he did an experiment with spirogyra where he essentially, without the technology we had today, made a pretty good action spectrum. And what he did was he put a filament of an algae here, and then he created a scattered spectrum of light. I think he used a prism. And then he put aerobic bacteria in there. And then wherever he saw a lot of colonies like this, he knew that there was a lot of action going on. And it made a remarkably similar shape to the actual action spectrum we got from a spectrophotometer nowadays. So yeah, basically just where there was aerobic bacteria, there was photosynthesis happening because oxygen was being released, and then those bacteria used that oxygen to grow. So yeah, that was just a pretty cool experiment, um, with pretty early techniques to figure out an action spectrum. All right, so here's just more about pigments. So chlorophyll A is a blue-green pigment. It's our main chlorophyll that we're, thinking of when we think photosynthesis. It's used by all photosynthetic eukaryotes and also by cyanobacteria. So in sulfur bacteria, they like to use bacteriochlorophyll. That's for purple bacteria, and it's blue-green. And green sulfur bacteria use a green chlorobium chlorophyll. And then there's some cyanobacteria that can survive in conditions where there's a lot of far red light instead of, the red light that's usually preferred by plants. And cyanobacteria, and they use a special chlorophyll called chlorophyll F, which is yellow. Some accessory pigments, which are just other pigments that whose main job isn't like absorbing light to do photosynthesis. A lot of times they have like protective uses, like they absorb other wavelengths that might harm the plant, or they kind of broaden the spectrum that you're absorbing from. So chlorophyll B is one of them. It's an olive green color. It's found in plants, green algae, and euglenoid algae. Chlorophyll C is green and it's found in algae. Carotenoids are red, orange, and yellow, and they are antioxidants, and they're used for photoprotection, which is protection against excess light. And they're found in all chloroplasts and in cyanobacteria. Phycobilins are found in cyanobacteria and red algae, and they can be different colors. So yeah, now we're on to the concept of excitation. So light, if you'll remember, it has a certain amount of energy, and that is due to its frequency or wavelength. It can dislodge or excite electrons, but in order to do that, it has to be of an exact special wavelength that matches that electron. And then when the electron is excited and it quote unquote falls, it releases energy. And if you just have this electron isolated, you're going to release some heat. And if you isolate chlorophyll and you put it in, a flask, it's going to fluoresce as well. So yeah. So if you think about say you have like a car outside in the heat and you touch the hood and it's hot, that's because the light from the sun excited the electrons in the hood of your car, and when they fell down, they made heat. So yeah. However, in plants, they use something called resonance energy transfer, where the energy from that electron falling, instead of making heat or light, gets transferred to another chlorophyll molecule, and it makes a chain like that until it reaches eventually here, where the electron is transferred to an electron acceptor, and that leaves an electron hole in the excited chlorophyll, and that makes kind of like, it's called an electron hole because it's like missing an electron, and then that's really just a redox reaction because there's a transfer of an electron. So yeah, here's a diagram just showing that. Okay, so photosynthesis is organized in photosystems, which are found in the thylakoid membrane, and essentially it just does those things I was telling you about earlier. We have a photon strike and excite an electron in a chlorophyll molecule, and then that energy gets passed on through chlorophyll molecules until finally they reach a special pair of chlorophyll A molecules in the middle of our photosystem, and that electron, that excited electron, gets transferred to what we call a primary electron acceptor, and that leaves an electron hole in these chlorophyll A molecules. So yeah. Some more information about our photosystem. So in our photosystem 2, which is our first photosystem, it was named in the order of discovery, not necessarily in the order things go through them in photosynthesis. It's called P680 because that is the wavelength of light that it would take to excite these central molecules, central chlorophyll molecules, and it's the strongest biological oxidizing agent known to man. And photosystem 1 has P700, so a slightly different wavelength. All right. Now let's talk about linear electron flow. So this is what you're probably going to think of when you think about photosynthesis. So we've got our two photosystems, first photosystem 2 and then photosystem 1. Our first step is light coming in, exciting an electron in this chlorophyll. That excited energy gets passed on until these pair of special chlorophyll molecules. And then there, the excited electron is going to be given to the primary electron acceptor. And now that's going to leave an electron hole in these chlorophyll molecules. So what's going to happen is that an enzyme splits H2O into, the two protons and then half of an O2 molecule, so an oxygen, but in the normal form, in a cell, it would exist as O2. It just spontaneously combines with another oxygen from a water molecule that was just split. And we also get two electrons. And what they're going to do is they're going to plug that electron hole. And then now the electron in our primary electron acceptor can move on to the electron transport chain. So in the electron transport chain, it's going to lose energy. And the chain leverages this energy to make ATP. We'll go over the specifics of how ATP is made in linear electron flow a little bit later. It goes through these parts of the electron transport chain, plastoquinone, cytochrome complex, and plastocyanin, and eventually it ends up here. So let's look at what happens here. In photosystem 1, largely the same thing happens. Light excites, an electron in a chlorophyll molecule. That energy gets passed on to these two special chlorophyll molecules, and then an electron there jumps up to the primary electron acceptor, and now we have an electron hole. And instead of plugging that electron hole the same way we did using electrons from water in photosystem 2, that electron from photosystem 2 that is out of energy, that is what comes and plugs the electron hole here. So now our primary electron acceptor gives our electron to ferredoxin right here. And those electrons go to NADP plus reductase, which turns NADP plus some hydrogen ions from outside the thylakoids in the stroma to NADPH. So you'll notice that in this process, a lot was done with hydrogen ions that we didn't really talk about. So over here, when we split water, these hydrogen ions stay inside the thylakoid. And when we're going down the electron transport chain, the energy from the electrons is used to pump hydrogen ions into the thylakoids. So we're gaining more and more hydrogen ions inside the thylakoids on this side. And then lastly, when we're making NADPH, we're taking a hydrogen ion from outside from the stroma. So what we've done is we've created a very deep gradient of hydrogen ions. There's a lot inside the thylakoids, and there's comparatively a lot less in the stroma outside the thylakoids. So that's going to help us make ATP. We'll get into that in, I believe, one more slide, but that's important to keep in mind. And then cyclic electron flow is an alternate method of electron flow. It's present in a lot of organisms, and a lot of people theorize it might be just kind of an evolutionary leftover, but it also might have like photoprotective effects. So essentially, largely the same as linear electron flow, except for when it reaches ferredoxin, instead of going to NADP plus reductase and having the electrons stay in NADPH, they get shuffled back to the cytochrome complex where they just keep on pumping those hydrogen ions on the inside and causing production of ATP. So yeah, photophosphorylation is how we're actually going to use that hydrogen ion gradient to make ATP. So if you remember from the cellular respiration lesson, we had oxidative phosphorylation. Well, in a lot of ways, it's the same. We're using that hydrogen ion or that proton gradient, and we're feeding that through ATP synthase, which makes ATP from all the hydrogen ions flowing through. So here's just some more information. The stroma's pH is about 8 and the thylakoids is about 5. And then I mentioned this earlier, but making NADPH also contributed to making that gradient. So yeah, we're just keeping a lot of hydrogens over here, and we're moving as many of the hydrogens here inside or removing them as we can. So yeah, hydrogen ions are going to flow through ATP synthase, and that makes ATP. All right, so now onto the Calvin cycle. We've made lots of NADPH and lots of ATP in the light reactions, and we're finally ready to turn those things into some sugar that we can use. All right, so in the Calvin cycle, there are 3 phases, and these are carbon fixation, reduction, and regeneration of the CO2 acceptor, or RuBP. So overall in the Calvin cycle, what we're doing is we're taking CO2 and we're making this sugar called G3P, which we can turn into glucose. So when we're looking at this cycle, we're going to be looking at what happens to 3 molecules of CO2 that are entering at the same time. So the first thing that happens in carbon fixation is that each of these CO2 molecules, the carbon, they contain 1 carbon, and then each of the molecules are added to a sugar called RuBP with 5 carbons by an enzyme named Rubisco, which is believed to be, I think, the most plentiful protein in the world. And we get a intermediate that is 6 carbons, but it's very unstable, so it breaks into 3 carbon sugars called 3-phosphoglycerate. So what we're going to do is we're going to spend 6 ATP, and we'll phosphorylate that first carbon, and it becomes 1,3-bisphosphoglycerate. Then we'll spend some NADPH. Now we're in our reduction phase, so we're going to spend some of that, and we're going to reduce our sugar. I believe we reduce the carboxyl groups. I'm not very sure on that, but anyways, we reduce the sugar, and one of the phosphate groups also comes off, and then we get what we call G3P. Now this is really nice. We have 6 G3Ps now, but we can't keep all of them because we put in 3 RuBPs for our 3 molecules of CO2, and each of those has 5 carbons, so we have to return 15 carbons to the cycle, or else it wouldn't be sustainable anymore. So we have 18 carbons, so one of our G3Ps we got just out of these CO2 carbons, so we're going to set that one aside. We can keep that one, but 5 of them are going to go back again into the cycle, into the regeneration phase, and essentially what this phase does is just turn it back into that RuBP, which can be inserted in the cycle again. So yeah, here's just some analysis. To make 1 molecule of G3P, you needed 3 CO2s to provide the carbons, 9 ATPs, and 6 NADPHs. All right, now part 5 is alternate mechanisms to photosynthesis. So photorespiration is the reason why we need alternate mechanisms to photosynthesis sometimes. So rubisco is not very good because it tends to have a higher affinity for oxygen instead of CO2. And in situations where plants are encountering a lot of oxygen being let in, that can be really bad because we put oxygen into the Calvin cycle instead of CO2, and that just wastes a bunch of resources and doesn't make anything useful. And that's especially problematic for C3 plants, which are, very big group of plants that we see every day, rice, wheat, and soybeans. It can reduce agricultural productivity of them by 50% because you're wasting a bunch of resources that we made in our light reactions, and you're not really doing anything. You're not making any sugars that can be used for energy, really. You're just wasting energy and resources. So yeah, we want to prevent these, which is why we have two mechanisms, the first one of which is C4 photosynthesis. So this involves mesophyll and bundle sheath cells in our plant, in our leaves specifically. So mesophyll cells are these cells right here. They do the majority of the photosynthesis, and then bundle sheath cells are around the xylem and phloem of the leaf. So it's used in C4 plants like sugarcane, corn, and grasses. You'll notice that these plants tend to be in drier, more arid areas. And the reason for that is photorespiration is especially bad in those drier, hotter areas because the plant doesn't want to open its stomata to get good gas exchange. So what happens is that the O2 Oh yeah, and that's because it's really hot, so the plant loses a lot of water, which is obviously bad. You don't want to be losing too much water. So what happens is that O2 produced in the light reactions gets trapped inside the leaves, inside the plant, and then that just makes it easier for rubisco to use oxygen instead of CO2 in the Calvin cycle. But yeah, that explains that. And then in the C4 pathway, basically what happens is CO2 comes in, and PEP carboxylase, an enzyme, adds it to PEP to make oxaloacetate that can be converted into malate or other organic acids, and that's brought into our bundle sheath cells. All of that happened in the mesophyll cell, and the CO2 is released again where it undergoes the Calvin cycle. So after the CO2 is released, we're left with a pyruvate, which goes back into our mesophyll cell, and an ATP is spent to turn it back into PEP. So since we're spending an ATP here, bundle sheath cells actually sometimes use cyclic photosynthesis to basically replenish that ATP that you're using. So that's another function of cyclic photosynthesis or cyclic electron flow that can be used in plants. Okay, so CAM, also called, it stands for Crassulacean acid metabolism. This type, it's not separating, the Calvin cycle with oxygen by space the way our C4 photosynthesis did. It uses time instead. It all happens in our mesophyll cells. It's used by pineapples, cacti, and other succulents like aloe and jade plants. So, plants that are usually in the desert. In fact, the name Crassulacean acid metabolism, that comes from a certain type of succulent, I believe, that uses it. So yeah, if you look at it, it's very much similar to C4 photosynthesis. We have our PEP, which becomes oxaloacetate with our CO2, turns into malate or other organic acids, CO2 released at night, where it's safer to open the stomata, and PEP gets returned. So largely the same process as C4 photosynthesis, just temporally separated instead of spatially. So yeah, these two are very important to remember, as well as what type of plants like to use these two. You know, C4 is a lot of the grasses, and then CAM is more of like the cacti and the succulents, stuff you find in the desert, you know? So yeah, that's it for today. Thank you so much for, watching this video. I really hope it helped. If it did, please leave a comment or a like and subscribe for more content. Thank you.